Okay, ladies and gentlemen, I would like to welcome everyone to our session, Next Gen Therapies. So, what I can say, it is my great honor and genuine pleasure to welcome Paweł Przewięźlikowski, CEO and founder of Ryvu Therapeutics, co-founder of Cellvita and RDGen. I will not list his professional and personal achievements. You can check it easily on different websites. It's publicly available. Instead, I would like to share my personal story about Paweł. I remember the very first day that I met him in Warsaw. We sat together over coffee, and he shared his bold vision of building one of the first biotech companies in Poland. A company dedicated to discovering first or best-in-class novel therapies in oncology. It was December 2007. Can you believe it? 18 years ago. And that single conversation opened a completely new chapter for me. From that moment, I began to immerse myself in biotechnology, exploring new therapies and modalities. And during that time, Paweł entrusted me a crucial mission to bring the first scientific director and key scientists to small, ambitious, and yes, admittedly risky company based in Poland, in Kraków, which at that time the company employed around maybe 15, 20 people in total. And, we did it. We did it together. We attracted outstanding scientists from abroad who later became leaders in their field and key contributors to the small molecule programs that ultimately reached clinical trials. It's a huge milestone. Years later, when I founded my own company, ExecMind, Paweł and Cellvita became our very first client in biotech. Once again, he placed his trust in me, and we delivered. It is truly moving for me that after all these years, now I have the privilege of welcoming to this stage a remarkable leader, someone deeply and truly committed to the growth of the Polish biotech sector, someone whose vision and resilience continue to inspire me. Paweł, the stage is yours.
Thank you, Karolina. Some things have changed, but you look exactly the same like 18 years ago. So thank you very much for the introduction. So I would like to tell you a few words about, the story of our most important drug, romaciclib, which originated in Kraków many years ago. It will be a story of, of discovery, of development, and hopefully some vision for the future of the program. A few words about what Ryvu currently is. It's a company that emerged as after the split of Cellvita to the therapeutic part, which is Ryvu Therapeutics, and contract research services part, which is called Cellvita. In the process, we also spun out RDGen, which is the largest AI drug discovery CRO in this part of the world. So Ryvu has two drugs in the clinic, romaciclib and daposertib, both invented a few kilometers from here at Jagiellonian Center of Innovation and developed by Ryvu team. We have about 200 people currently. Unfortunately, we had to lay off some colleagues a year ago because of the difficult situation in the biotech sector. But let's hope that, this biotech winter is starting to be over and soon, the Polish biotech industry will start growing again. We are also publicly listed on the Warsaw Stock Exchange. Our most important programs are in oncohematology, in life-threatening diseases, deadly diseases, such as acute myeloid leukemia, myelofibrosis, or some more benign disorders such as lower-risk myelodysplastic syndrome. We are also starting a new study in medulloblastoma, which is a children's brain cancer. Behind this, we have about 100 people in discovery of our novel programs. Sometimes we collaborate with partners. The most important partners for us are Menarini, Biotech, and Exelixis. So let's go to the beginning of the history of romaciclib, which originally was called Cell 120. Then, after we changed the name from Cellvita to Ryvu, it started to be called Ryvu 120, and now it's called romaciclib to honor the main father of this drug, also who Karolina knows very well, Dr. Tomasz Rzymski, which obviously Rzymski means Roman in Polish, so that's why we named the drug after him. So the target for romaciclib is cyclin-dependent kinase 8, CDK8, which, unlike other CDKs, is not a part of, it doesn't regulate the cell cycle, but regulates RNA transcription. So its mechanism of action is different from CDK2, 4, 6, 7, 9. A lot of this has had to be discovered internally at Ryvu Labs in Kraków, and also through different academic collaborations. We learned a lot also from third-party research at various universities from the United States, Poland, and Austria, for example. Romaciclib was discovered in our labs at Bobrzyńskiego Street. And this is actually the first laboratory notebook, as you can see, Cellvita, and the pages describing the synthesis and the first experiments on romaciclib. So please remember, well, if you discover, you have to properly document it. If we didn't have this laboratory notebook, someone could, for example, challenge our patent applications. But we have all the proper documentation, and that's why we know exactly when this drug was first time synthesized and what was the story that had to be properly documented after. This is the team that worked on romaciclib. Many of them are still at Ryvu, and this is a structure of the drug which is quite interesting because not very frequently we have bromines in drugs. Some people thought that it's not a classical structure, but it's quite inventive, and this structure has served us well. It's not very toxic. It's working in multiple indications. So the chemistry that we started based on this drug was quite efficient. As you can see, there were many examples. So in order to discover a drug, you frequently have to synthesize hundreds or even sometimes thousands of analogs, closely related compounds, in order to arrive at the molecule that will be of the final interest. So obviously, some of the synthesis that we were making in this project was on a small scale, but obviously later we had to move to larger scale, and we needed to have proper labs for larger scale synthesis. But of course, when you go to the clinic, it's no longer possible to synthesize the drug at a place like our labs at Sternbacher or JCI. You have to go to a real drug factory, and currently we manufacture most of the substance at Polpharma in Starogard Gdański, and we also collaborate with other international CROs to make capsules out of this drug. But for the in vitro experiments, for the in vivo experiments, we could synthesize the sufficient amount of the compounds at our labs in Kraków. This is actually, to the left, it's the first time we tested it in vitro. and we have the pictures of this experiment. Obviously, everything had to be properly documented, so we have an archive of all the information. And for the first time, we saw that we have efficacy. The compound was tested in many concentrations, and at the larger concentrations, we were killing cancer cells completely. That was the start, but obviously, after we did we performed the in vitro studies, we put 12 different compounds to PK studies, about 52 different in vivo experiments in various tumor models, in solid tumors, such as colorectal cancer, in various lymphomas, leukemias, breast cancer, and other indications. And finally, when we had selected our clinical candidate, we had to perform a lot of toxicology experiments at various concentrations, various dosing schemes in order to arrive at the optimum balance between efficacy and safety. And originally, many people don't remember this, but it was originally a solid tumor program because CDK8 is overexpressed in multiple cancers, and the most pronounced overexpression is actually in colorectal cancer. So we were thinking we would be developing a solid tumor program, but then it turned out that the activity in solid tumors, especially in colorectal cancer, is not that strong, but we have much better efficacy in leukemia, especially in a form of leukemia called acute myeloid leukemia, which is a disease that you just go, it's very lethal. You can just walk around on the street, then feel unwell. They take you to a hospital, and if there is no proper treatment, you can die in 3 days. That's why it's called acute. Many patients obviously survive this treatment, but for many of them, there are no therapeutic options, especially if they relapse after the initial success of the treatment. So this publication about AML put us into a certain direction, which we are more or less exploring right now, because most of the activities around romaciclib are focused on hematologic malignancies. Obviously, a lot of additional experiments, selectivity profile, optimization of chemical structure, understanding of the binding, so that we can really find out which compounds should be synthesized, which will be kind of more toxic, and then a lot of different acute myeloid leukemia cells to show the efficacy of the program and compare where we have the best responders and some non-responders. And obviously, we are also using not just Ryvu 120, but also senexin, which was another CDK inhibitor, to confirm that these effects are not just compound-specific, but also protein-specific. So then we were also testing it in combinations with other drugs that are frequently used in AML treatment, and we understood the mechanism of action. Actually, romaciclib has a very interesting mechanism of action because it kills cancer cells, but it also forces leukemic or pre-leukemic stem cells into differentiation. So we are able to restore normal erythropoiesis with this drug in different settings. Okay, this is the timeline of discovery. So we started in 2011. It's already 14 years ago. Then we had, the first in vitro data, the first in vivo data, the original patent application from 2013. Then a collaboration with the Center of Oncology in Warsaw. We moved to AML focus on AML maybe after 3-4 years into this project. We put this drug to IND-enabling studies in 2017, and we dosed the first patient in AML in 2019, and the first patient in solid tumors in 2021. So it's a long journey, too long. I wish it was much faster, but we had a lot of issues on the way. The most important was lack of money. Sometimes we were struggling to have $20,000 for additional experiment. We had lack of expertise, sometimes bad luck. Like, for example, we had a clinical hold that was imposed after a patient died from pancreatitis. and there was a suspicion that romaciclib is causing pancreatitis, but only after a few months did we find out that the patient had a pre-existing pancreatitis before entering the study, and she shouldn't have gone on the trial, but the hospital didn't tell us at the time. But then, because of this, we lost about a year or even more, because we had to remove the suspicion of pancreatitic toxicity of romaciclib. So such stories, you are like Sisyphus, you are pushing a stone up the hill, and then the stone is rolling constantly, and you have to have a lot of resilience to bring this to fruition. Probably with the current resources, this story would have been much shorter. And I hope that we learned from this, and many Polish biotech companies are also learning with their first projects. And I hope that then, as a local industry, will be moving these drugs faster. The world record, probably for a first-in-class drug of this that took us about 8 years, will be 3-4 years, and the record from the first patient in to an approval for cancer drugs is about 3 years. So definitely this timescale should be compressed, and especially the Chinese companies are very good at compressing these timelines. In Poland, unfortunately, and generally in the European Union, we have stupid regulations, especially regarding animal experiments. Sometimes it takes us 6 months to get a permit for an experiment, and people are dying because someone is protecting this mice from this mice. So if we don't change such rules, we will not have drug discovery in Europe, and we'll be just kind of closing our eyes and doing everything in China. Okay, that's probably one of the most important letters that we received. Approval to start a clinical study that we got from the US FDA. The first clinical trial was in the United States, and romaciclib became a clinical stage drug. Okay, what we think about romaciclib right now. So this is the mechanism of action, CDK8/19 inhibition. CDK19 is a paralog of CDK8, and 4 different disease development paths for the program: acute myeloid leukemia, myelofibrosis, which is a disease where patients' blood cells are produced not just in the bone marrow, but for example in the spleen. So the spleen, instead of being whatever the size of a pear, becomes the size of whatever a grapefruit or even bigger, sometimes 2 liters. And the patient is very exhausted, they lose weight, and so on and so on, and it can transform to acute myeloid leukemia. Lower-risk myelodysplastic syndrome is a disease in which basically you have anemia, and you have to have transfusions, and when you exhaust options, you may also transfer to high-risk MDS, to acute myeloid leukemia. So our job is to prevent these transfusions, restore the normal hemoglobin levels. And in solid tumors, we are currently focused mainly on this brain cancer, medulloblastoma. This is our development timeline. So currently we are finishing the exploratory part of the study. And if everything goes right, we'll be able to start the approval process for romaciclib sometimes at the end of 2028. And the drug will not be approved earlier than in 2029. So imagine it will take at least 18 years from the initial synthesis to an approval. Definitely a very long story, and it's not something that we are very proud of. But this is how we could do it. Certainly, you all can improve on us. Okay, the studies that we are now conducting is the most important study is River 81. It's in a combination with a drug that is already approved in acute myeloid leukemia called venetoclax. In this study, we dosed more than 50 patients already with our drug. And this is the data that we presented. So essentially, the most important data was here. So essentially, in this particular cohort, we had 43% response rate, and duration of response was something like 2-3 months. But all the patients were ongoing, and in December we'll inform what happened to these patients. On the patients who are put in this setting that have exhausted the therapeutic option of venetoclax azacitidine treatment have an average overall survival of 2.3 months. So anything better than 2 months is already a big achievement. And obviously the response rate that we are observing here is quite encouraging. Let's hope that the data will be further validating this approach. In myelofibrosis, we dosed more than 20 patients already at different sites. And this is the data that we presented in June at the European Hematology Association conference. This is the swimmer's plot. These are spleen volume reductions after 12 weeks of treatment. And these are so-called total symptom score improvements after the first 12 weeks of treatment. So we could see that also in myelofibrosis, we observed initial signs of efficacy. And I hope that this could be confirmed, deepened, and expanded with our data readouts in December. Okay, this is how, for example, the total symptom scores look like. So here we had a patient who was suffering from whatever belly pain, night sweats, fatigue and bone pain and everything. These are the typical myelofibrosis scores. And after 3 months of treatment, these symptoms have completely resolved. So this person felt normal. And this is what we would like to achieve in other patients. So definitely, romaciclib is efficacious as it shows signs of efficacy. The big question is if it's efficacious frequently enough and strongly enough to be an approved drug. That's the question for the next years. Okay, if we are successful, then obviously these are large markets, hundreds of millions of dollars, sometimes billions of dollars. So, but the biggest thing for us is just to get it approved, and even a small market will be enough to pay for the expenses, for risk, and we'll be very proud of bringing this drug to patients worldwide. This is the study that I'm particularly fond of. It was proposed by Professor Rajiv Vipakar from Colorado Anschutz Center. And then we got together with Professor Bożena Dębowska-Wagińska from Centrum Zdrowia Dziecka in Warsaw, Children's Memorial Hospital. And we will be using romaciclib based on Professor Vipakar's idea in children with medulloblastoma. Average age of diagnosis 3 years. 30% of children die. Those who survive have been exposed through this treatment to radiation, chemotherapy. They develop all kinds of disorders. So a very serious unmet medical need. So thanks to the grant from Medical Research Agency, we'll be testing romaciclib in these children. And I hope that their life will be better because of this. Let's see. But this is a long-term project. It's non-commercial. Maybe we'll get the data in 4 or 5 years from now. Okay, so this is the global footprint of romaciclib. We dose patients in the United States, in Europe, more than 150 hospital contracts. It's probably one of the largest, if not the largest, innovative clinical trial in Poland. And let's hope that this whole effort, operating effort, will come to some fruition. Our key advisors, in various disorders, we collaborate really with top experts. Most of these people, Courtney DiNardo is #2 in acute myeloid leukemia in the world. Eitan Steen, #6. This is also top 10, top 20 in other indications. So really, I wish that we had more Polish people as the most important key opinion leaders, but I hope that we are learning together and you will see some Polish names in the next years. Maybe some people who will become famous because of romaciclib. I'm also very proud of the access that we are giving to Polish patients, thanks to grants that we receive, thanks to fantastic academic and medical collaborations. We have a lot of sites in Poland that we are using for various clinical studies, and these patients really have a difference and have access to better treatment. Interestingly, something that we should just kind of be we have to understand is sometimes it's you are not your prophet in your own city, because probably Kraków was the 50th city in the world when we could dose some patients. Simply no other cities around the world and in Poland were more open to welcome our clinical trials. And then I was joking that if we start dosing patients in Kraków, then we have really succeeded globally. Okay, so our plans for romaciclib, I hope that the data in AML myofibrosis will support so-called accelerated development pathway, that pivotal studies may initiate in 2026 and approval process in 2028, and very favorable commercial conditions. These are rare diseases that have premium pricing, so I hope that if we succeed, then we will just have some return on investment of this about $50 million that this project has cost so far. So to end the story, this is the story of discovery from the biological concept, chemical synthesis, in vitro, in vivo testing, lots of solid tumor, lots of trials in hematology, in solid tumors, the ongoing trials that are going to read out over the next months, all our phase 2 studies, and the future strategic goal, an approval in global and enabling access to this drug for hopefully thousands of patients worldwide. We are certainly very focused on this. Thank you very much.
Thank you, Paweł. Brilliant story. And so we keep our fingers crossed for the positive readouts and for the first drug in oncology from Ryvu. So congratulations, and we stay tuned. All right, so it's time for our next speaker, or generally speakers, because we have a pretty intensive time right now. So I would like to welcome Dr. Anna Bratek-Skicki of the Jerzy Haber Instytut of Katalysis and Surface Chemistry, Polish Academy of Science. Dr. Anna Bratek-Skicki studies protein adsorption, molecular interactions, mechanism of stress granule formation linked to neurodegenerative diseases, and your work combines physical chemistry, yes, colloidal science, and advanced biophysical modeling. Is that right? That's correct. Okay. Okay, so it's yours.
So, good morning, ladies and gentlemen. First of all, I would like to thank the organizer for giving me the opportunity to be here and present my research. And today I would like to talk about amyotrophic lateral sclerosis and the challenges that are ahead of us in understanding the mechanism that is behind this disease. So what is ALS? So amyotrophic lateral sclerosis is a rare progressive neurological disease that affects the nerve cells in brain and spinal cord, responsible for controlling the voluntary muscles movement. And eventually it will affect muscles which are responsible for movement, speech, eating, and breathing. So there is no cure for ALS at the moment. And the danger is very serious because the average survival time after diagnosis is about 2 to 5 years. One of the exceptions was Professor Stephen Hawking, who lived with this disease for over 50 years. But at the moment, there is speculation if it was really ALS or a combination of few neurological diseases. So where we should search for the roots of this disease? So of course, we should do it in the basic structural and functional units of living organism, which is the cells. So the cell contains various organelles, which can be divided into two groups: the membrane-bound organelles, which are listed here, but also membrane-less organelles. And one of them are stress granules, which are formed in different. They are different stress granules depending on the stress conditions. So stress granules are formed when the cells face the stress, and under physiological conditions, it's a normal process which prevents the cell from dying. But under certain circumstances, when the stress is prolonged, it might lead to diseases. So these stress granules contain many different molecules, and one of them are RNA-binding proteins. And misregulation in this RNA-binding proteins is implicated in many diseases, including amyotrophic lateral sclerosis. So what is also important, that maybe I will come back for a moment, that these membraneless organelles are formed via a process called liquid-liquid phase separation, where the homogeneous milieu in the presence of stimuli forms two-phase system, one high-density state when we observe the droplets, and low-density state. So when the nuclei, the small droplets, is being formed, it evolves over time. And here you have the three main paths reported in the literature. But in general, it consists in two stages: the nucleation growth, where we observe the liquid-like structures, droplets, and maturation, which leads to formation of aggregates, which are present in neurons, and they are responsible for their death. So why we are interested in these processes? First of all, we would like to understand the mechanisms of their formation in order to design and work on appropriate treatments. We would like to know also which species are toxic to the cells. Is it monomeric version, oligomeric, or maybe bigger aggregates? Bigger aggregates, we already know that they are very toxic to the cells. So what we are doing in our laboratory? In our laboratory, we express, purify, and characterize these proteins, and we study the three processes: nucleation, liquid-liquid phase separation, and aggregation. So here I'm presenting typical results that we obtained for one of the proteins, HNLNP2, involved in formation of stress granules. We proved using different methods that these proteins exist in form of oligomers. Here we have results achieved by Brownian microscopy, atomic force microscopy, and also cross-linking. The size of those oligomers depends from protein to protein, but for sure they don't exist in form of monomers. So in our papers, we work also on the second phase transition, liquid-liquid phase separation. And in 2022, we reported in our paper that studying that the liquid-liquid phase separation of protein involved in ALS is very sensitive to experimental conditions. We show that working with protein that it's, I'm talking about, of course, in vitro measurements, experiments that are working with for example, urea, in the presence of detergent with proteins that contain solubility tag, which improves the stability of the proteins, might lead to very false results. We propose method in which we are able to work in near-native conditions and get the true results about the mechanism involved in LLPS. So what else we are doing? We are testing in our lab series of different compounds in order to modulate those three processes: nucleation, LLPS, and aggregation. Here I'm showing you some results for our compound that is at the moment confidential, that leads to stopping the liquid-liquid phase separation in vitro. We also perform some experiments under dry condition using atomic force microscopy, where we prove that by adding this compound, we significantly reduce size of those oligomers, and then the liquid-liquid phase separation doesn't occur, doesn't happen. So, of course, many different studies is needed to prove efficiency of this compound. And what is also important to highlight that this compound is already approved by FDA agency and is used for different disease. So if it works in real system, that could be easier a way to put him in the market. So what are we looking for to collaborate with? So at the moment, we are looking for undergraduate student, PhD student, to make the work faster. We also search for scientists and laboratory that could help us with theoretical modeling, who has expertise with cell models expressing proteins involved in ALS, animal model, but also drug screening. So at this moment, I would like to thank those people and organizations who made possible to conduct this research I'm presenting, and thank you for your attention.
Thank you. That was very, very interesting. So do we have some time for questions? Do we have any questions from the audience? No, any? Okay.
I got a question.
Oh,
Yes.
How did you select this compound that you showed that you are testing?
I worked with this compound years ago during my PhD. And knowing the interaction with other compounds, I thought that might work for proteins, especially taking into account the chemical formula and also the presence of certain amino acids in the proteins. So I think it was a guess. So I tried and it worked. Yes.
Congratulations.
But we have many other compounds that we are trying with different proteins. Those results which I presented here were performed only for one protein. I came to Poland just 1 year ago and I just started this research, so I really need to speed up.
okay. Thank you so much once more. And if you want to reach out, Dr. Anna, so you know how she looks like right now. So thank you once more. Okay, so now it's the time to welcome Dr. Agnieszka Tomala from Krakow University of Technology. Dr. Tomala is a materials scientist specializing in biotribology. So can you please explain us later what does it mean exactly? And surface engineering biomaterials for regenerative medicine. Okay, so the stage is yours.
Thank you very much for your introduction. I'm representing Krakow University of Technology, and the work that I will present today is titanium hydroxyapatite composites with MXN coatings for long life osteoconductive implants. This work is done within Opus Lab project with Wave Unisono program from National Science Center, and this is done in a collaboration with Vienna University of Technology and University of Łódź, the biology department, and this is the entire team that we are working with. Of course, we have an increased number of orthopedic implants that are needed every year due to the aging of the society. We mostly focus on the heavily loaded joints like a hip, knee, or shoulder. And the materials that are used for the replacements are metal, ceramics, or like ultra-high molecular weight polyethylene. For the arthritic knee, the surfaces become irregular and they are rough. That's why there is a high friction in between the knee and the joint. That's why the replacement surgery is we are replacing the cartilage surfaces with the smooth surfaces that are correcting the deformities and reducing the stiffness. In our work, we are actually developing we are working with both metal and ceramics. We are combining metal and ceramics together, and we are developing the composites made of titanium and hydroxyapatite. The hydroxyapatite is actually a natural component of our bones, so it's very easily osseointegrating with our body. Moreover, we are using laser surface texturing to to open the underlying porosity. Such a laser pockets also serve us as a storage sites for the material for the ExecMind that we are applying on the top of the laser texture. With this method, we are able to produce such a gradient titanium hub components with the ExecMind top layer on the top. Our partner from Vienna University of Technology is developing the ExecMind and coating the substrates. The ExecMinds are produced by electrochemical etching method, and my colleague today at the Green LifeScience will talk about the production methodology. By the spray coating, we are covering the surface that was the laser texture with the ExecMinds. For the developing of the composites, we are using the powders of the porophore, of the hydroxyapatite and titanium that are mixed in the planetary ball mill with the zirconia oxide grinding jar and the same material for the boards to keep the clear chemistry. The pressing with the cold isostatic pressing and the sintering is actually the most challenging process because the most important in case of our material is to keep the hub crystallographic structure the same before sintering and after sintering. Our first attitude was the sintering in the vacuum furnace. However, it turned out that the vacuum is causing the dehydration and dehydroxylation of the hydroxyapatite. So now we developed the method to how to sinter our composite to keep the same crystallographic structure of hub, so it does not decompose during the process of the material preparation. And this is done by using a protective also zirconia oxide yttria stabilized nanopowder. Also use such insist plates, which are causing that we are able to produce such a gradient structure. The gradient structure is composed of the pure titanium that is keeping actually the mechanical properties, and the titanium hub part, which is actually mimicking the trabecular part of the bone. The roughness of the surface changes with the cross section. It's very, very smooth underneath. So this cortical, so as I said, it's mimicking the cortical part of the bone, the lower part, and the roughness is below is around 0.20 micrometers, and above the spongy part, the trabecular part, is the roughness is much higher than. Here is 6, but it's actually ranging from 6 to 10 micrometers. Our surfaces from the top point of view are very rigid and corrugated, which is actually promote the cells to osteointegrate. Moreover, on the top of the surfaces, we have such a cauliflower structure that are our hub particles, and this we can prove by the presence of the calcium and phosphate that indicates this presence of hydroxyapatite. Moreover, our surfaces are very porous, and that's why we are using additionally a porophore to increase this porosity. The porosity depends on the composition, is varying from 50 to 25%, and the pore size are in the range. The most of the pores are located from 10 to 100 microns, and there are less pores that are much higher than 100 microns, 200, even up to 300 micrometers. Moreover, the surfaces are very hydrophilic. They are actually super hydrophilic because they are soaking all the fluid that is applied on the top of the surface. This is particularly good for them also as a drug carrier. We also did incubation and biological activity study, and we have shown that with the incubation, the pH is very stable, especially in simulated body fluids. It's varying a little bit for the artificial saliva, but it's actually related with the apatite formation of the top of the surface. This is actually the first indicator that our surfaces are very osteo-integrated, because during the incubation process that takes 28 days, we have a very thick layer of apatite on the top of the surface, and this is also can be shown that all the entire titanium peak, the titanium substrate is covered with the calcium and phosphate. And that is actually suggesting us that the apatite form on the surface. Moreover, the cytotoxicity was measured on the powders and on the materials for the mouse fibroblast and human osteoblast, and it shows no cytotoxicity. First, for the powders, we actually got very good results for the electrochemical produced MXNs and the green MXNs, and also the MXNs that are applied on the top of the surfaces also do not show any cytotoxicity. Moreover, the cell viability is over 100%. We also performed tribological performance in order to see how such an implant will behave in our body during the movement. Here we observed that the MXN is causing the huge reduction of friction. The friction is very low in this case. We did it for different combination of the materials with the zirconia ball and the steel ball. And we observed that with the zirconia combination of the material, this is giving very low friction and no significant wear was observed. This is particularly related to the smearing of the MXNs on the top of the surface, as it is shown here. Summarizing all this research, so we have developed gradient titanium hub MXN composite biomaterials that are mimicking the cortical and trabecular part of the bone. We maintain the same hub crystallographic integrity through the optimized sintering process. Our pore size are in the range from 50 to 25%, from 10 to 100 microns, that are ideal for the bone cell infiltration and regeneration. Moreover, the in vitro bioactivity with formation of apatite layer after incubation. We see that the cell viability exceed 100% for both of the cells that we have tested, and we observed the unique delamination of MXN effect that is observed in the friction contact zone. For the collaboration offer, we are mainly now focused to try our biomaterials in animals, in rats, rabbits, and bigger animal, starting with the goat, and to observe the regeneration capacity, inflammation assessment, and the response of them on the implant side. Thank you very much for your attention, and many thanks for my team.
Thank you. You are doing incredible things. So do you have any questions? Okay, so Dr. Anna will be also reachable during this conference. So thank you so much once more. So right now we are going slightly to business side. So let me introduce and welcome to this stage Jakub Gauza from Voxel. Jakub is a radiochemist developing next generation radiopharmaceuticals with a focus on copper-64. And this is for combined diagnostic and therapeutic applications, improving accuracy and monitoring in modern nuclear medicine. The floor is yours.
Thank you. Hello, everyone. My name is Jakub Gauza, and I'm a radiochemist in Vauxhall at Center for Radiopharmaceutical Production in Krakow. Today, I will briefly talk about copper-64 and this role PET diagnostic as well as the cancer therapy. Let's start, however, with how PET works. The PET scan utilize positron emitting during the beta plus decay, decay of radioactive substance. The fundamental principle is the process is annihilation process. A positron upon contact in a positron upon contact with in the electron undergoes immediate annihilation. This result in the creation two gamma radiation quanta that travel in opposite direction at the angle at 180 degrees. The patient is administered a radiotracer, most often via intravenous injection. For example, the substance 18-FDG is a glucose dilative that travel to cancer tumors whose cells have demand for glucose. The PET scanner is a ring composed, one might say, of many Geiger counters, which, by receiving the emitted gamma radiation, allow for the 3D visualization of the area where the specific radiopharmaceutical accumulates. The primary application of PET, for obvious reason, includes use in oncology. We can detect cancer and also monitor its treatment process. Besides this, we can examine the functioning of organs. For example, studying the hair function. Additionally, we can monitor and treat neurological disease such as Alzheimer's and Parkinson's. The most commonly used radiopharmaceutical is 18-FDG. However, an increased number of radionuclides are emerging in cancer diagnostic, such as gallium-68 and copper-64. For the diagnosis of disease, we can use technetium-99m. Liver disorders such as Wilson's disease can be monitored using copper-64. And the next one is using iodine 123 in, for example, IFP-CT molecule. We can examine neurological disease such as Alzheimer's and Parkinson's. Okay, take a look. Use the radioisotope copper-64. This long half-life offers several logistical benefits, and ultimately can mean being able to most easily shuttle patients. But one of the main features of copper-64 is a short positron range. with a short positron range, the annihilation point occurs close to the atomic nucleus. Signals are emitted closer to the emission source, which can lead to high spatial resolution. A smaller positron range allows for a more precise visualization of the cancer location and help detect additional smaller object. The radio metal copper-64, for example, possesses such properties with a positron range about 3 times smaller than that of gallium-68. This allows for seeing a greater amount of detail the scan. Copper-64 is a unique radioisotope because it undergoes beta- decay, beta+ decay, and electron capture. Its positron emission is suitable in PET imaging, allowing precise diagnosis and tumor localization. At the same time, its beta- particles can deliver therapeutic radiation directly to cancer cells. This dual capability, both diagnostic and therapeutic, makes copper-64 a prime example for theranostic agent. It has been shown that copper-64 can be excellent diagnostic agent for detecting prostate cancer as well as neuroendocrine tumors. Who we are? As the Voxel Group, we specialize in the production and utilization of radiopharmaceuticals. We also developing new radiotracers. Our company possesses the necessary infrastructure. We have 2 cyclotrons in Krakow and Warsaw, and in the future we will have a 3rd cyclotron in the new radiopharmaceutical production center in Katowice. We currently have 4 of our own radiopharmaceuticals. Our R&D department is also working on the development of the radionuclides. We also own diagnostic center Trove in Poland. We can fulfill orders of radiopharmaceuticals, precursor radiopharmaceuticals, and cyclotron-produced radionuclides. We develop new technologies, are open to new challenges. Why collaborate with us? Because we have access to cyclotron. We produced isotopes and radiopharmaceuticals. We have experience in supporting drug development and regulatory compliance, and a wide range of service for life science research. We also conduct a number of scientific studies within the framework of researchers' project and grants. Current work is being carried out as part the project przeprowadzanie prac badawczo-rozwojowych produktu radiofarmaceutycznego do diagnostyki raka prostaty i guzów neuroendokrynnych. This project financed by the Medical Research Agency. In addition, we are in the process of registering clinical trials using copper-68 data talk. The study has already been assigned number, and its registration is moving forward. And that's all from my side. Thank you, everyone. Sorry. Thank you for your attention, and we. Thank you very much.
Thank you so much. Thank you. Do you have any questions about the radioisotopes or diagnostic? Okay, so Jakub also will be reachable during the whole conference. Do you stay tonight?
No.
No, just okay. So you need to be hurried.
Travel to Warsaw.
To Warsaw.
To Warsaw.
Okay. So have a nice trip back home. All right. Thank you so much. So our next speaker and presenter is Dr. Jacek Olczak, founder and CEO of Ascentia and co-founder of Onco R&D and Molecure now. A very experienced organic medicinal chemist with more than 25 years of experience in drug discovery. He has led numerous multidisciplinary programs, developed novel synthetic methods, co-authored over 30 publications, 12 patents, and I believe you have your own small molecules in clinical trials.
Talking about it.
Okay, perfect.
Okay. So thank you very much for introduction. I would like to also thank organizing committee for giving me opportunity to introduce my third company I have founded in my life already, Ascentia. We are basically custom research organization, meaning organization, company who helps other companies, teams to deliver drugs. We are based in Łódź, in a technological park called Bio Park at Dibuła. First slides about founding team. Me, as you can recognize, Dr. Marzena Mazur, Director of Chemistry, Dr. Damian Jacenik, Director of Biology, the youngest guy in the founding team, Mariusz Milewski, Computational Chemist, and two members of Scientific Advisory Board, Professor Beata Kolesińska, who is an expert in peptide synthesis and natural product synthesis, and Professor Jakub Gołąb, who is an expert in immuno-oncology. And Dr. Aleksandra Wodarczyk, Chief Strategy of Business Development Officer, who is also with me at this conference. So what is our claim to fame? These are two molecules that are currently in clinical trials that have been discovered in my team at my previous company, Molecure. One is chitinase inhibitor, now in phase 2, in the indication of sarcoidosis, which is rare disease based on fibrotic etiology, and the second one, arginase inhibitor, which is now in phase 1/2A clinical trial in immuno-oncology. So these are part of our publications that we have published over the years working on those two compounds. You can find it through the Web of Science or Google Scholar. I'm showing it just to show you that in terms of small molecule drug discovery, we do know what we are doing, and we can help you to discover your drugs and develop your drugs. So in every drug discovery program based on aiming at small molecule drug, there is always a moment when you need some compound to be synthesized, whether some molecular probe or some stuff which is difficult to source, difficult to synthesize, or is too expensive, or you don't know how to get it. Just come along, talk to us, and we will help you. So these are our technical capabilities that we currently have in Bio Park, but that's simply organic chemistry laboratory. We have also biological testing capabilities, from testing compounds in relatively simple biological tests like enzymes or receptors through the test on cells, cytotoxicity profiling, and permeability measurement, and so on and so on. We have also access to the much more sophisticated techniques through the collaboration with Medical University of Łódź. There is a lot of equipment that is waiting to be used. So like methods like PCR or flow cytometry or Western blotting. Everything could be arranged through the collaboration with academia in Łódź. We are also using the electronic lab books, which is nowadays it is obvious, but for some reason, especially for customers who order larger projects, it is of value because the customer has a direct insight into what is going on in their projects without need of scanning laboratory notebooks, pages, and sending them along. So, summing up, we are not just the executors who are doing what we are told, but we are scientific experts. We provide our scientific insight based on long-time experience. So we had a lot of also international collaborations in the previous companies that we were running before even Molecure. And we respect your IP, and we are here. We talk Polish, we talk English. So I encourage everyone to contact us. So we are looking for collaborations of different kinds. We are open for discussion with startups, spin-offs, or small teams, biotech companies, also academic teams. There is a lot of interesting projects running in academia. So yeah, beside the drug discovery, also other sectors are in the area of our interest, which is cosmetics, agrochemicals, and everything which is connected to life sciences. So that's it. Thank you.
Yeah. Thank you. Thank you so much. So I have a question, if I may. So what are your current plans for next year? I hope it will be more optimistic than last one for the biotech industry.
Well, we are in Kraków, where Cellvita was born, and I do envy the success of Cellvita and Ryvu has achieved. And I think that it is possible to repeat such a success in my home city, Łódź. And I think that there is a lot of opportunity here in Poland to grow the company like Ascentia to much larger volume. So besides serving customers, of course, we have our own ideas to develop our own drugs. However, here I'm presenting my company as a CRO because I need the positive cash flow to feed my company, right? But beside the cash flow, yes, we do have our own drug discovery plans, but it is not the time to talk about them right now.
Okay, so next year?
Hopefully.
All right. Thank you so much. This was Dr. Olczak, so he's also reachable during the whole event. Thank you.
Thank you.
All right, so it's time for our next speaker. Now we are slightly getting moved back to academia with business applications. So let's welcome to the stage Julia Kaniuka, a student from biomedical engineering at AGH University of Krakow, University of Science and Technology. Julia develops innovative actuator and measurement systems using shape memory alloys with applications in medical devices, robotics, and prosthetics. Okay, so here we go.
Okay, hello everyone. Thank you very much for your kind introduction. My name is Julia, and today I would like to present a project about integrated actuator and measurement system based on nitinol fiber. This work explores how a single nitinol wire can act both as an actuator and as a sensor. Firstly, I would like to shortly describe what exactly is a nitinol, then show the motivations behind this project, and describe the system architecture and show key measurements and characteristics, followed by a description of our control algorithms and experimental results. So firstly, what exactly is a nitinol? It's an SMA material, which means it's a shape memory alloy. And what's extraordinary about this material is its ability to go back to its remembered shape after a deformation. And it is also made alloy from a nickel and titanium. And as you can see on this simple diagram, when nitinol is in its original shape in lower temperature, it can be easily deformed, for example, bent, and it will hold this deformed shape. But once it's heated, it reverts back to its original remembered shape in this higher temperature. And also during these differences in this material, it also changes its resistance, and that's why it could be also used as a sensor. Nitinol is already widely used in medicine, for example, as a self-expanding stents, or in orthodontics as an arch wires or retainers. And you can trust me because I have something like that for my teeth too right now. But not only as a material, because we can easily heat the nitinol, for example, by electric current. So we can apply it to some electronic circuits, to electronic devices, and it could be used, for example, to move the artificial fingers, or also to move a flexible tip of an endoscope, for example. So the goal of this project was to develop an integrated actuator and measurement system using the nitinol to track its displacement via electrical resistance, and to control its non-renal behavior, which I will show you after a few moments. But firstly, I would like to describe the system architecture. So we used a nitinol wire connected between two terminals. T1 was fixed and T2 was movable. To the T2, we applied a non-elastic cord that holds 0.1 kilogram load. So when the electric current flows through the wire, it shrinks. And we also applied a magnetic encoder that measures the displacement of a nitinol. We also have an electric circuit that measures the voltage and current of the nitinol wire and calculates it to the resistance in the real time. And all the control algorithm was applied in Arduino microcontroller. And here we can see some measured characteristics. First, we recorded the displacement of in the function of current, then a voltage drop on the nitinol wire, and it was then calculated to the resistance. And we can see on this chart a typical nitinol behavior, the hysteresis loop, which means that the wire behaves differently when it's cooled or where it's heated. And this is the main problem if we want to precisely control the movement of this wire. Here we can see the resistance and displacements as a function of voltage control signal. And as you can see, they are also affected by a hysteresis. by considering the direction of changes, rising or falling, we can estimate the displacement, but the estimation, just from the voltage control isn't very stable, because even if we have a voltage control on the same level, the temperature of the wire can still change after a time, so it also affects the resistance. So we should consider to put two curves to the control algorithm, and we chose the voltage displacement curve, and that generates the main control signal, and also the resistance displacement data that verifies and corrects the position of the wire. And we applied a light wire control algorithm on the Arduino that measures the desired direction, measures the actual resistance in real time, and uses also additional support signals that are increasing or decreasing the voltage value that is depending on the measured resistance. So, for example, if it's too low, then the control algorithm applies the SU signal, U from up, and increase the voltage control, so the needle wire can reach the desired displacement. And we tested this control circuit by applying a start-step signal asking the wire to move from positions of 1 millimeter to the 9 millimeters. And as we can see, it reached the target positions quite good. And the statistical analysis of a few runs of the device showed a relatively narrow tolerance band, which means that the system has relatively good repeatability under stable conditions, and can be good for the future work. So to conclude, we designed a control system by just using a simple Arduino micro controller without using, for example, additional PID controllers. In the future, the signal parameters could be optimized, and the system is scalable to multi-actuator setups, so it could be used in the future to design, for example, as I said before, the prosthetic hand or the endoscope tip, or also my next step in this project is to design a surgical needle with actuators made from the needle that can bend into different direction and generate a precise motion. And this research was supported by the project Support for Students in Enhancing Their Competencies and Skills under European Funds for Social Development Program. And thank you very for your attention.
Thank you so much. It was a great presentation. So I have one question. So what kind of collaboration are you looking for at the moment?
So at the moment we're looking for collaboration to apply the system to design the more medical devices, for example, like in the guided needle, surgical needle, or in the endoscope tip, because it's very important to have very precisely medical devices. And as I know, for example, in endoscope, now it isn't very precise, so we want to develop it. Yeah.
Okay. So wish you all the best. Good luck with your project, and thank you.
Thank you.
Applause. So right now, let's welcome to the stage Dagmara Słota from Krakow University of Technology. Dagmara is a materials researcher from Krakow, working on gold polymer nanocarriers for targeted anti-cancer drug delivery and imaging. And your focus is on designing more precise, less toxic and highly traceable cancer therapies. Okay, so welcome.
Thank you. Okay, hello, ladies and gentlemen. It's my great pleasure to represent today the Krakow University of Technology, Faculty of Materials Engineering and Physics. And today I would like to tell you a little bit more about gold polymeric nanocarriers for targeted therapies of cancer disease. And my goal is to introduce you to our idea to bring this technology closer to you and maybe encourage you to cooperate with us in the future. So, as you probably know, cancer remains one of the greatest medical challenges of our time. It is estimated that in Poland alone, around 100,000 people die every year with the cancer, and around 1.70 million patients live with the cancer daily. Also, it is estimated that around more or less 170,000 new patients are diagnosed each year. So as you can see, the problem is really big. The problem is growing every year. And that's why we need good therapy. And also the current therapies, they lack the precision. So the anti-cancer drugs, they really often affect the healthy tissues. Also, the doctors, they do not have the control over the drug distribution in the body, so they are not sure if the drug is reaching the tumor site in the body. And also there is, of course, cancer cell resistance, so that's why we need we need better, more safe and more targeted therapies in the medicine right now. So before I introduce you to our idea, let me just tell you that the final beneficiary of this technology are of course, oncology patients that required better, new, and safer technology and safer treatments. And patients' needs also. Those patients also, they will gain an access to significantly more safe and less toxic therapies. With our idea, we hope that doctors, they will gain an access to a dual function therapeutic tool that enables both the treatment, but also what's the most important, the ability to track the distribution route of the drug in a patient's body using CT imaging. And thinking about future, we really hope that potential pharmaceutical and biotechnological companies, R&D institutions, they will gain an access to our know-how, to our idea and this innovative technology. So about the idea itself, sorry for rozwiązanie, which means the idea, the solution. It's over missing, of course. Anyway, the whole idea is, as I already told you the carrier of anti-cancer drug. And the carrier itself, it's made out of biocompatible polymers. It's a mix of 3 polymers, polyvinylpyrrolidone, polyethylene glycol, and polyhydroxybutyl. And that's the base, that's the matrix of our invention. And this polymer base is enriched with inclusive complex made out of gold nanoparticles. Anti-cancer drug with this capecitabine is a drug that is used in medical practice with breast cancer. And that's all is connected also with, combined also with beta-cyclodextrin. And the idea is to release this inclusive complex from the carrier to the body. And here's where magic happens, because the gold nanoparticles, they have those ability to reflect the light. So basically, this complex with these gold nanoparticles can be observed using CT imaging as a whiter spots on the pictures, which means that the doctors, they can track the distribution route in the body. And also, just to inform you, because it's not that obvious, but the gold in nanometric size, gold nanoparticles, they have this red rubish color, so that's why the color of the carrier is pink. And all of that, in our opinion, it means precision, safety, possibility to personalize the dose of the drug. Also, it will enable the distribution, to track the distribution if the drug is reaching the tumor site. And it's all made out in environmentally friendly and eco-friendly technology. Just a couple information about the researches that we conducted so far without going too much into details. We optimized the production, the synthesis of the gold nanoparticles, of course. We checked the adhesion, the attachment of those inclusive complex to the surface of the carrier. We also check the surface roughness and the sorption capacity. As the sorption capacity, the swelling of the material is one of the way to deliver the drug in vivo conditions. Also, we of course measure the release of the drug of the capacity bin in vitro studies, and last but not least, we confirm the biocompatibility using cell lines. And what we are looking for, of course, we are looking for collaboration, because it's just the idea, and it's not just the idea, because we already conducted some studies. We already have a patent application, and we thinking about extending into a PCT procedure, because so far it's only in Polish patent application. However, we would like to conduct more research. We would like to extend this technology. We hope to conduct research with cancer cell lines, and of course preliminary in vivo animal studies. And from our side, we can offer you an access to our laboratory, to our equipment, and of course to this new technology, to our know-how, to our experience and our knowledge. And this is our team. Me and Karina, we are main researchers in this technology, and our leader is Professor Agnieszka Sobczak-Kupiec. She's the leader of this project, but also the leader of the whole team of biomaterials at my department. Bożena Tyliszczak and Agnieszka Tomala, those are postdocs and professors, our mentor in this idea, who help us to grow this technology. Agnieszka, she presented before me, and Karina, she's presenting right after me, so I encourage you to stay a little bit longer on this session. If you have any question, you can contact me via LinkedIn, and if you have any questions, you can also ask them right now or at the lunch. I'm open for collaboration and for questions. Thank you.
Thank you so much. Brilliant idea. Congratulations. So do you plan to spin off this technology to set up a new entity?
We'll see. We think about it. We have this InTech company, InTech institution at Krakow University of Technology. So I think we have to collaborate with them and then think about the future of this innovation.
Yeah, perfect solution. It's really encouraging to see such brilliant minds located here in Krakow and in our universities. So thank you.
Thank you.
All right. So as you mentioned, your colleague, so it is also my pleasure to introduce Karolina Niziołek, PhD student, also from Krakow University of Technology. This is a very productive academia. So researching, Karolina conducts research about gradient polymer ceramic biomaterials for osteochondral tissue regeneration. And your work integrates materials engineering and regenerative medicine to support bone cartilage repair. Okay, the stage is yours. Yeah, applause.
Thank you very much. Okay. Good morning. My name is Karolina and I'm a PhD student at Krakow University of Technology within the Faculty of Material Engineering and Physics. Today I'm honored to present our research focused on gradient biomaterials for functional osteochondral interface regeneration. Osteochondral defects are still one of the biggest challenges in orthopedics. Even though implants are getting better, they still cannot recreate the bone cartilage junction, which is why we fail quickly. Because of this mismatch, tissue do not connect properly, implants fail too early, and the patients often need additional surgeries. This problem is becoming more important as more people suffer from degeneration, injuries, or damage caused by overuse. What's more, traditional implants and cements are poor tissue integration, risk of many complications. That is why we need new biomaterials, safer, long-lasting, and able to support natural tissue regeneration. Our solution is follows a biomimetic approach, which means that we imitate the natural structure of cartilage and bone interface. Market data, which we can see at the right, also shows that demand for cartilage regeneration is growing fast and may nearly double by 2028. This shows that better solutions are needed as soon as possible. From both the clinical and patient's perspective, there are several important needs that we must consider. First, the need for effective regeneration of cartilage and bone defects. Current implants are often not durable. Second, the materials must be high biocompatibility, so they do not cause immune reaction or toxicity. Third, implants should be fit better with the body. And it's also important to ensure faster recovery and fewer post-operative complication for patients. Finally, the modern implants, modern medicine is moving toward personalized solutions, such as implants with many shapes or structures, often made, for example, for 3D printing or cross-linking methods. Our target groups include orthopedic and trauma hospitals and clinics, medtech and biotech companies, R&D units, and research centers. Ultimately, however, the main beneficiaries are patients suffering from joints defects. And our solution, the base of our materials is a scaffold made from polylactic acid, produced using FDA method. This structure gives our scaffold, our implant, good mechanical strength and a honeycomb shape, which allows to control porosity and helps cells attached. On the top of this scaffold, we added a layer of polymers such as polyvinylpyrrolidone, polyethylene glycol, and pullulans such as polysaccharide. Into these layers, we added the calcium phosphate particles. What is important in our solution that we add the particles in a controlled gradient. The highest amount in the part that should behave like bone, and the amount slowly decrease toward the cartilage-like part. This architecture mimics the natural structure of the injection bone and cartilage, gradually changing parameters of stiffness, porosity, and bioactivity. After the photocross-linking method, the whole multi-layer system becomes a stable gradient materials with mechanical and biological properties that we can adjust and need. And research results, we conduct several in vitro studies to evaluate cytotoxicity, cell adhesion, cell activity, and over biocompatibility of our materials. As you can see, indirect cytotoxicity was tested using two methods, one natural red uptake assay and XTT metabolic activity assay. In all tested sample, cell viability stay high, so it's good for our biomaterials. We also used scanning electron microscopy to observe how mouse osteoblast cells interact with our materials. The images show that cells attached well and spread on the surface and form normal extension. In another experiment, we tested proteoblast cells on our gradient materials for 14 days. The metabolic activity increased over time. Overall, these results showed that our gradient biomaterials support our cells adhesion and their growth. Together we achieve more, and in the future we plan to continue developing our project by combining our knowledge from materials science, clinical research, and biotechnology. We also plan to do our preclinical studies, in vivo tests on animal models, biocompatibility, and regeneration efficiency analysis. What's more, we want to go to participate in invention exhibition. What's more, we want to protect our invention by the patent protection at the Polish Patent Office, and also we plan to extension to the PCT procedure. Also, this research was funded in Wall by National Science Center with project hierarchical approaches for osteochondral tissue engineering. And this is my team, my dream team, where I worked. We together won 4 medals at innovation exhibition. Also, we had 1 patent application to the Polish Patent Office. And thank you for all for your attention. And that's it.
Yeah, thank you. You have brilliant innovations there at Krakow University of Technology. So congratulations to all the team. So thank you. I guess you're also reachable during the whole event. Okay, perfect. Perfect. So now, dear guests, let me introduce to the stage Łukasz Indyka, senior scientist from Selvita. Łukasz is a scientist from the medicinal chemistry. He also took lead in some research projects as principal scientist. And your core expertise is medicinal chemistry, synthesis of molecules. All right, the stage is yours.
Thanks a lot for your kind introduction. So I will talk today about the development of a platform for hits identification in antiviral drug discovery program. So I work currently as a senior scientist in chemistry department in Selvita, and I was involved in this research project as a principal investigator. So generally in Selvita, we work on commercial projects for external clients. So this one was a little exception because it was an internal research project that was meant to develop a portfolio of our services. So I will start first with the introduction of the background and the goals of the project. So the idea emerged at the beginning of COVID-19 pandemic. Which showed how important it is to have effective treatment for infectious diseases. Even now, after the pandemic, infectious diseases and especially viral diseases are still a serious health, social, and economical problem worldwide. Current therapies are very often limited and bring numerous side effects. That's why development of new effective therapies is still a very important matter. So in this regard, our project was meant to facilitate and speed up a discovery of new drugs that could be used as a potential treatment for infectious diseases. The main goal of the project was a synthesis of a focused library of compounds with potential antiviral properties that were designed to target viral proteases, which are the crucial enzymes in the viral life cycle. This library was a base for generation of a service platform that includes all required chemistry, biology, CADD, which means computer-aided drug design, and ADME tools for hits identification and development of novel antiviral therapies. So how the library compounds are designed? So the key part is difluoroketone group, as shown here in this slide. This is a warhead which is meant to covalently bind with the catalytic amino acid residues in active site of an inhibited enzyme. Most often, in case of proteases, this is a cysteine, serine, or threonine. A formation of covalent bond, first of all, blocks the enzyme activity, prevents the virus from processing its proteins, and stops viral replication at an early stage. So our warheads are based mostly on difluoroketone moiety, but this was also our starting point for further modification, which led us to a variety of warheads as depicted here in this small picture. They all possess fluorine atoms, so incorporation of fluorine enhances their electrophilic character, and this also affects its affinity toward catalytic residues in active sites. So the compounds that are included in the library are peptidomimetics. They can be classified as peptidomimetics, which means they are very structurally similar to peptides. And peptides are natural substrates for proteases. And this allows our compound to effectively bind and inhibit activity of proteases. So a little bit about methodology. So this synthetic scheme depicted here was one of the main methods that we used for incorporation of a difluoroketone group into our compounds. So I won't go into details because first of all, we don't have time, and also I think we don't have many organic chemists here. So the main point was that we were able to use this methodology for production of quite a lot of different warheads. And also it allowed us for further derivatization. We were able to obtain many interesting warheads with distinct properties. So all synthesized compounds were characterized by NMR and LCMS. And we decided also to test the activity of these compounds on biological assays against 5 different common proteases, namely 3CL SARS-CoV-1, 3CL SARS-CoV-2, HIV, HCV, and papain-like SARS-CoV-2. The biological activity was assessed as a % of inhibition at a given concentration, but also for the most active compounds, we decided to determine their IC50 values. Selected representatives also went through more detailed testing that includes determination of basic ADME parameters such as metabolic stability, kinetic solubility, or chrome-log-D, which is a lipophilicity parameter. So the results, we were able to obtain over 200 unique full-size compounds that were screened against 5 different proteases and more than 800 molecular fragments, which are suitable for a fragment-based drug discovery approach. Over almost 25% of tested compounds showed a significant level of inhibition against one of the tested proteases. So an example of the compound that was tested is depicted here. So we have a warhead, which is supposed to covalently bind the catalytic residue in the active site. And this compound can be considered as a hit molecule against SARS-CoV-2 protease. And also, ADME profiling showed very satisfactory values across all tested parameters. So these values are similar to what literature presents for similarly structurized compounds. And application, so currently this platform, the developed platform, is offered to pharmaceutical companies and scientific institutions, or basically anyone who is interested in the development of novel antiviral drugs and therapies. And the created compound library is can be used for high-throughput screening and is a part of Cellvita's broader portfolio of screening services. All synthesized compounds are stored in such vials as shown here, barcoded vials, and can be used, are ready basically for any downstream testing. For more details about compound library that are offered by our company, you can visit this link below. And so this was our team. So from left to right, Patrycja Morowiec-Kucharska, who was a biologist, Sylwia Wójtowicz, ADME specialist, Olga Krzysztyńska-Kuleta, also a biologist, Georgi Kaczkowski, who was a project leader and concept founder, Amanda Ferreira, a senior scientist in chemistry department, me, Matija Piro, chemist, and Wiaczesław Stepanenko, who is a lead scientist in our department. So that's all. Thanks for your attention.
Thank you so much. So do we have any questions to Łukasz? Okay. So, Łukasz, are you available during the whole event today?
I think I will be.
Okay. Okay. Perfect. So, thank you so much. And right now we are getting to the last two presentations. Both of them are coming from Krakow University of Technology. So, let me welcome to the stage Dr. Katarzyna Hraźna, assistant professor, working on biopolymers, biomaterials, polymer-based scaffolds for peripheral nerve and spinal cord regeneration. And she has led multiple national research projects and received several scientific awards. So, Professor, stage is yours.
Thank you.
Yeah.
Thank you for your kind introduction. Ladies and gentlemen, it is honor to present our results from research is focused on preparation materials for neuronal tissue regeneration. So I would like to start to focus on the problem which is connected to use implants and biomaterials. So due to intensification of human life and prolongation of average human life expectancy, the more research is focusing on the develop on new implants materials. This is also connected to a lot of side effects, which is related to use traditional autogenic, allogenic, and xenogenic transplant. This is why doctors are looking for the new materials which can used in the surgical procedures. Particular attention is put on the use of materials for peripheral and spinal cord regeneration. As we can see in the figure, the value market is very high and is expected to grow in the following years. So, our group is focusing on design and characteristic of new scaffolds materials. Before me, Dagmara, Karina, and Agnieszka talk something about materials for bone, osteochondral and regeneration, and also about preparation of novel drug carriers for anti-cancer substance. But now I will talk about very specific properties which is desirable in the case of materials for neuronal tissue regeneration. First of all, when we think about preparation of these materials, we want to have procedure which can help us easy prepare of these materials and prepare with low cost. After that, we want to have also several different properties such as lack of toxicity, citable mechanical properties, macroporosity, and bioactivity. We want also have materials which have biomimetic properties and that can stimulate the regeneration of nerve. So, in our project, we developed different kind of materials, the film and porous one, and these materials are composites. So briefly, we're mixing different compounds. First was polymers, mainly polyesters, polyhydroxyalkanoates, and small amount of polycaprolactone. We also used inorganic nanoparticles, nanofiller, which was MXN, that have very interesting properties in the case of neuronal tissue regeneration. First of all, they have conductive properties. They can stimulate nerve cells to first to adhere to surface. After that, to proliferation and differentiation and also maturation. This nanofiller has also antibacterial properties, which is very important for materials using in surgical procedures. We use also active substance. I will talk about CBD oil. This substance is very interesting because have several different properties. For example, analgesic, anti-inflammatory, antioxidant, and what is the most important, neuroprotective. We now are working on the preparation of tubes with specified dimension, diameter and length to meet the requirements of individual patients. So now we can look at the key features of the materials. First, in our test, we got the results that our materials has appropriate degradation rate and are characterized by absence of harmful products. We also perform a lot of different biological tests. We established that our materials are characterized by lack of toxicity, can stimulate adhesion of neuronal cells to the surface, can stimulate proliferation, what we can see here, differentiation and maturation of cells. We also confirmed this using confocal imaging, which was performed at the Instytut of Pharmacology, Polish Academy of Science. We also established that we can incorporate to our ExecMind other active substance, not only CBD oil. And in our test, we also got the results that our materials have antibacterial properties. They are effective against microorganisms, both gram-negative and gram-positive. And in the test in which we used macrophages, we established that our materials have anti-inflammatory properties. So our materials have a lot of advantage. We can prepare materials with which will be customized for a specific patients in the case of length, diameter, or incorporate of active substance. We believe that we can help in faster recovery, and we can inhibit. We can get materials with minimal risk of rejection after implantation. So our materials can be attractive for example, people which suffer from different injuries of the nervous system, for example, following accidents, spinal cord injuries. We believe also that our materials can find application in surgical procedures and can be interested for biotechnology and pharmaceutical companies. And now we are working on preparation of materials using different additive manufacturing method. We also are ready to start our next research, which will be focused on combination of materials engineering techniques, biological tests, and AI models for the preparation of materials for spinal cord regeneration. I know that this is hard subject, but we are ready, and we are also looking for potential collaboration. Both universities, scientific institutes, and business. This cooperation can be, for example, can be focused on using a more advanced characteristic technique. And at the end, I would like to acknowledge funding institution, our brilliant biomaterials team, and collaborator for help in preparation and in characterization of our materials. And thank you for your attention.
Yeah. Thank you. Thank you so much. So if I may, I have one question. So do you plan to license this technology to find a bigger medtech partner who has a portfolio in cord spinal technologies?
This is a very interesting question. At this moment, we of course, we thought about this, but at this moment, we think that we should
focus on the research?
Yes,
or?
yes.
Okay.
Thank you.
All right, great. Thank you so much. So we have one speaker left, Dominika Trager from, again, Krakow University of Technology. Dominika is a materials engineering student, and she contributes to the development of hybrid ceramic polymer composites for next generation 3D printed bone reconstruction materials with enhanced integration and mechanical stability. All right.
Thank you so much.
Yeah, applause, of course.
Thank you so much. So, first of all, I would like to say that today I would like to share a concept of our huge project that I am able to be part of. And I would also like to say that my part of the project is also my bachelor thesis degree that I'm going to hopefully defend it in the 2 months. So let's start it. But first of all, please take a look at this diagram and present it, and please considering what factor maybe connect the 4 areas presented: the AI technology, 3D printing, the increased need of the surgery, or the increased numbers of the chronic disease. Of course, the answer is always, it depends. But if we simplify it to just the medical implants, we can see the clear answer. And their answer is increased value in the markets, increased growth and the value. And these 4 areas represented is just the key driven to its growth. But shall we take a look at the numbers? And the numbers are telling us that in this year, the 3D printing medical implants market size, it's about $2.66 billion, and it's predicted to grow by $9.81 billion by 2033, with a CAGR of nearly 60%. But the growth couldn't be possible without the new solutions, without the new technologies. So right now we can see on the market that the personalized bone implants are slowly become a reality. And we can see that the number of biocompatible approved materials for printing is growing and multiplying. We can just exchange like a polycaprolactinate material or polylactic acid material or even polyurethane. And I think the major advance for this material on the market is that they can provide a valuable alternative for traditional transplant grafts. However, there are always 2 sides of the story. So if we look at the specific, we can choose polylactic acid. We can see that it has some problems, even though it's really popular. If we're looking at the mechanical sides of it, like mechanical characteristic, we can see that the polylactide has really big problem with a low strength at its solid state of the molting stage. We can see that it's really breathless, and there is of course a problem with the inflammatory effect when the material is in contact with the tissue. So we ask ourselves, with all of this knowledge, what can we do, and can we even do anything to provide a difference? And the answer is yes. But we have to change our approach to the problem. We have to change our approach to the materials, even change the all materials by itself. And in our laboratory, in the Krakow University of the Technology, we have developed a method for producing and formulating a composite material for 3D printing, designing to offer a new improved functionalities compared to commercially used materials. One of the main goals is obviously bioactivity, meaning the material will provide good terms for the cells to grow, for cells to proliferate. The material is also intended to be biomimetic, which is the structure of the material should be similar to our natural bone structure. Of course, if we're talking about the structure, we have to talk about the porosity of the material, and that also is the thing of our design, because of the pore, the blood vessels can grow into our material and provide the good quality of the regenerate process. And of course, the material cannot degrade over time. So, to just conclude the concept, our concept behind this project is to build an innovative combination of a carefully selected and processed polymer, which use polyether ether ketone, with the addition of bioactive ceramic. The polymer phase provides really good mechanical characteristic, while the ceramic phase provides the good bioactivity and good biocompatibility of the material. So as a result, we obtained the material in a form of granules that is going to be a future implant. And as we all know, it is notably direct granule printing that has recently gained significant popularity, which means that in many cases we don't have to process our material twice. We just have a granule and then we can just print it all out. Right now, we are in the process of verifying our components and our material under the near-real operational conditions. In vitro and in vivo tests on the animal models are still lying ahead. That's why we are looking for the collaboration and partnership, and we are looking from the research institutions, clinical organization, and industry. We believe that by working together, we can make splendid material even faster and better. Of course, none project could be done without proper funding, and the name of our project is Bioactive Composite Granules with Potential for 3D Printing, and the project is carried out within the LEADER program. And finally, I would like you to introduce our team. A few of them you saw before me. And I also marked our leader, Ms. Dagmara Słota, she was before me. And if you have any questions, or maybe you would like to start cooperating with us in this project, or maybe something else, feel welcome to join us after this, to approach us after this session. We will talk and discuss about future cooperation. Thank you for your time and attention.
Thank you so much, Dominika. It's really brilliant to hear that today we had many brilliant scientists. And who said that scientists only work in lab? It's not anymore the truth. So thank you so much for your attendance today. Thank you to all our speakers. And so what can I say more? I hope that we see you next year in next edition. And I see that during the last years, we've made, as a biotech industry, including academia, a huge progress in making further development of different projects, starting from drug discovery and development, ending up to biomaterials, which is so needed for the advancements in medical technologies. So thank you once more, and enjoy the rest of the day and the evening, and enjoy also shaping and creating the whole life sciences industry. So the word is yours. Thank you.